Welcome back to the deep dive. Today we're opening up a file that is an absolute staple in medical microbiology. But honestly, the more you look at it, the more it feels like this bizarre biological riddle.
Oh, it completely is. It's a massive riddle.
Right. We're tackling the hepatitis viruses today. And,
Yeah.
I say riddle because usually when we group viruses together, like the herpes viruses or, say, the retroviruses, they're family.
Yeah, you expect a family resemblance.
Exactly. They look alike, they act alike, they have similar DNA or RNA structures.
Oh.
But here we have this absolute rogues gallery of completely unrelated characters.
A total mix.
We've got hepatitis A through E plus G. And they just couldn't be more different. Different families, different genomes. Some are DNA, some are RNA, completely different shapes.
Right.
Yet they all share this singular obsessive tropism for one specific organ, which is the liver. It feels like a heist movie where a safecracker, a demolition expert, and a hacker who've literally never met all decide to rob the exact same bank on the same day.
I love that. That is a fantastic analogy to start with. And it really gets to the heart of what we call the hepatotropic paradox.
Hepatotropic paradox.
Yeah. Why the liver?
Yeah.
Right. It isn't just a coincidence. Think about what the liver actually does. It's the body's primary filter. It receives this massive blood supply straight from the gut via the portal vein.
Okay.
So whether a virus enters through the mouth and gut or directly into the bloodstream through a needle, it is almost inevitably going to wash up in the shores of the liver.
So the liver is basically the net that just catches everything, including these totally unrelated viruses.
Precisely. And while these viruses are genetically distinct, the way the liver responds to them is remarkably uniform. That's why clinically they can look so similar on the surface to you as a practitioner.
Which makes it confusing.
Very. So today we're diving deep into Murray's Medical Microbiology, Chapter 55. That'll be our structural spine. But because we really want to get into the nitty-gritty of why these viruses are so tough, we're also going to layer in some crucial virology details from Jawitz, Chapter 35.
Perfect. So our mission today is to decode this alphabet soup for you. We've got A, B, C, D, E, and G. And if you're listening right now, maybe you're prepping for boards, maybe you just want to understand the lab work better. This topic is notorious for being overwhelming.
Oh, the serology charts alone.
They look like hieroglyphics. You've got varying cancer risks, different transmission routes. It's just a lot to keep straight.
It is a lot, but I promise there is a logic to it. Before we get into the specific viruses, we need to set the stage with a couple of broad mental anchors.
Okay, lay 'em on me.
The first is a classic mnemonic that has saved countless medical students on exams. Vowels affect the bowels.
Vowels affect the bowels. Okay, I like that. So we're talking about hepatitis A and hepatitis E.
Exactly. A and E are transmitted via the fecal-oral route. They enter through the ingestion of contaminated food or water.
Right.
And crucially, because of this route and their biology, they tend to cause acute self-limiting disease. They hit hard, they cause a lot of chaos, and then they leave. They generally do not linger to cause chronic infection.
Okay, so if the vowels are the gut punchers that hit and run, what about the consonants? B, C, and D.
The consonants B, C, and D are bloodborne. You can associate the C with continuous or chronic.
Oh, that's good. C for chronic.
Right. These are transmitted via blood, sexual contact, or vertical transmission from mother to child. And crucially, these are the ones that have the potential to set up shop for the long haul.
So they don't just hit and run.
No, they cause chronic infection, which is where the real long-term danger of cirrhosis and primary hepatocellular carcinoma comes from.
That is such a helpful divide. Vowels for bowels and acute, consonants for blood and chronic. Now, there's one more unifying concept we need to unpack before we meet the actual cast. You mentioned that the liver reacts the same way regardless of which virus it is.
It does.
So what is actually causing the damage? Is the virus getting inside the liver cells and blowing them up like a balloon until they pop?
That is the common assumption. We tend to think of viruses as elytic. They infect, replicate, burst the cell. But for the hepatitis viruses, that's generally incorrect. Really?
Yeah, these viruses are non-lytic. They can replicate inside a hepatocyte, a liver cell, without killing it directly.
Okay, wait. So if the virus isn't killing the cell, why does the patient turn yellow? Why does their liver fail?
It's collateral damage. From us.
Yeah.
From our own immune system.
Wow.
It's an immunopathogenic mechanism. The virus infects the hepatocyte, and the host's immune system, specifically the cytotoxic T cells, detects the intruder.
The T cells.
Right. The T cells realize the liver is compromised, and they just launch a scorched earth campaign. They attack and destroy the infected liver cells to stop the viral replication.
So going back to my bank robbery analogy, the bank isn't destroyed by the robbers, it's destroyed by the SWAT team trying to get the robbers out.
Exactly. The jaundice, the fever, the nausea, that is the SWAT team at work. That is the inflammation and necrosis caused by your own T cells.
That's wild.
It is. And this explains a fascinating clinical paradox. In patients with weak immune systems, or in neonates, you often see massive levels of virus in the blood, but very mild symptoms.
Because the SWAT team is asleep. Or just severely understaffed. If you don't fight back, you don't get the inflammation, but you also don't clear the virus. That's the trade-off.
That is a critical distinction to keep in mind. Okay, so we have our framework, vowels versus consonants, and it's the immune system causing the mess. Let's start at the beginning of the alphabet. Hepatitis A, the acute assassin.
Let's do it. Hepatitis A. This is a member of the picornavirus family, specifically the hepatovirus genus. Now let's break that name down. Pico means small, and RNA tells you the genome, so it's a tiny positive sense, single-stranded RNA virus.
And structurally, what are we looking at? Is it wearing a coat?
No coat. It is a naked capsid virus. And I want to pause here because this is a very high-yield concept for understanding transmission. In the viral world, being naked usually means you are incredibly tough.
Wait, that seems backward. Wouldn't an envelope or a coat protect you?
You would think so. But a viral envelope is made of lipids, fats. It's a membrane derived from the host cell. What destroys fats? Detergents, soaps, stomach acid, bile, drying out.
Oh,
If you strip the envelope off an enveloped virus, it dies. It's actually quite wimpy. It needs moisture.
but a naked capsid.
A naked capsid is just a tight protein shell. It's built like a tank. It doesn't need that oily layer. According to Jawitz, hepatitis A is stable at pH 1.
pH 1, that's pure stomach acid.
Exactly. That is why it can be transmitted fecal orally. You ingest it, it hits the acid bath of your stomach, and it just floats right through, laughing.
Unbelievable.
It's also resistant to detergents, solvents like ether, and it can withstand heat up to 60 degrees Celsius. You can dry it out on a surface, and it remains infectious for a week. Jawitz even notes it can survive in salt water or groundwater for months, even at negative 20 degrees Celsius for years.
That is terrifying. It's like the Terminator of viruses. It just doesn't stop. So if it's that tough, how do we actually kill it? Like if I wipe my counter with just standard soap and water, is that enough?
Probably not. Soap works great on enveloped viruses like COVID or influenza because it pops that lipid bubble. For hepatitis A, you need something that denatures protein. Chlorine. Bleach is the gold standard. Formalin works too. Autoclaving obviously works, but you can't autoclave your salad.
Right, right.
This environmental resilience is exactly why it causes outbreaks.
So let's talk about those outbreaks. You mentioned fecal oral, so contaminated water, dirty hands. But there's a specific food item that always pops up in the textbooks. The shellfish connection?
Ah, yes. Clams, oysters, mussels, the classic vector.
Why specifically shellfish? I mean, I assume we aren't talking about thoroughly cooked clams here.
Usually raw or undercooked, yes. But the reason shellfish are such efficient vectors comes down to their biology. They are filter feeders. A single oyster can filter up to 50 gallons of water a day. They're essentially biological vacuum cleaners.
50 gallons. That is a massive volume.
It is. Now imagine that oyster bed is near a sewage outlet, or just runoff from a heavy rain that overflowed a septic system somewhere. The water might have a very low concentration of virus, just dilute sewage. But the oyster is concentrating everything it finds. It doesn't just stick to the shell. The virus actually accumulates in the digestive glands of the shellfish.
So if you eat a raw oyster, you are eating the oyster's stomach contents.
Effectively, yes. You are ingesting a bioconcentrated viral pellet. And because of that naked capsid toughness we just discussed, a quick steam or a squeeze of lemon juice does absolutely nothing. You have to boil them thoroughly for several minutes to denature that capsid.
Note to self, thoroughly cook the clams. Now, aside from the seafood buffet, there's another major setting for hepatitis A transmission, right?
The daycare center.
Yes, the daycare vector. This ties back to the clinical presentation and what we call the iceberg effect.
Break that down for us. Why is it an iceberg? Because what you see, the jaundice, the sick patient, is just the tip. In children, especially those under 6 years old, hepatitis A is overwhelmingly asymptomatic or very, very mild. 90% of infected children won't turn yellow.
90%.
Yeah, they won't look like they have hepatitis. They might just have a runny nose, a little diarrhea, or low fever. They look like they have a standard daycare bug.
So they are the silent spreaders.
Precisely. They're shedding massive amounts of virus in their stool, but they don't look sick. The daycare workers change the diapers. Maybe hygiene isn't perfect. Remember, this virus laughs at simple hand washing sometimes, And then the virus spreads to the adults, the parents, or the staff.
And the adults don't get off so easy.
No. The immune response in adults is much more robust. 70 to 80% of adults will develop jaundice. They get the abrupt onset of fever, nausea, and that dark urine, which is the hallmark of liver dysfunction.
That is diabolical. The kid acts as the stealth bomber, flying under the radar, and the parents take the massive hit.
And the timeline makes it even trickier. Viral shedding in the stool peaks before the symptoms even appear. By the time the adult turns yellow and realizes they have hepatitis, they have already been shedding the virus for 2 weeks.
Wow.
They were most contagious when they felt perfectly fine.
That is a public health nightmare. But the good news is, A is for acute, right? It doesn't stay forever.
Correct. A is for acute, A is for away. There is no chronic carrier state for hepatitis A. You get it, you feel terrible for a few weeks, and then your immune system clears it. It does not lead to cirrhosis, and it does not lead to liver cancer. Once you recover, you have lifelong immunity.
And how do we prove that with a lab test? If a patient comes in yellow, how do I know it's A and not B or C?
Serology. You are looking for antibodies, specifically IgM anti-HAV.
Okay, let's practice this. IgM.
Think of IgM as the Marine, the first responder. If you see IgM anti-HAV, it means immediate acute infection. The battle is happening right now.
Yeah.
If you see Ig anti-HAV, think G for gone or grandmother. It means the infection is old history. It indicates immunity, either from past infection or vaccination.
Simple enough. IgM is, right now, IgG is history and prevention.
We have a very effective killed or inactivated vaccine. It's recommended for all children over 1 year of age and for travelers going to endemic areas. It has massively reduced the incidence of HAV in the developed world.
So hepatitis A, naked, tough, fecal, oral, shellfish, silent kids, acute only. Case closed.
Case closed.
All right, let's move to the heavyweight champion of the liver world. Hepatitis B, the complex chameleon. This feels like a huge step up in complexity.
It is. We are leaving the world of simple RNA viruses and entering a very strange territory. Hepatitis B is a hepatovirus. Hepa for liver, DNA for, well, DNA.
But this isn't your standard DNA virus, right? I was reading about the structure in Murray, and it seems like it uses decoys.
It absolutely does. This is one of the most fascinating survival strategies in all of virology. If you look at the blood of a highly infected person under an electron microscope, you don't just see one type of particle. You see 3 distinct shapes.
Okay, walk me through them.
First, you have the actual infectious virus, which we call the Dane particle. Jowett specifies this as 42 nanometers, the double-shelled sphere containing the DNA. This is the one that could actually infect a cell.
The real deal.
Exactly. But floating around it, outnumbering it by a huge margin, you see these smaller spheres and long filaments. These are roughly 22 nanometers.
And these smaller ones, are they infectious?
No. They are completely non-infectious. They are empty. They're composed entirely of the surface protein HBsAg, but they have no DNA inside. They're just empty shells.
So why does the virus make them? It seems like a massive waste of energy to build millions of empty houses.
It's military-grade deception. Imagine the host's immune system as a missile defense system. It sends out neutralizing antibodies, the missiles to stop the infection. The antibodies swarm the blood, looking for that surface protein to latch onto.
Okay, I'm tracking.
But the virus has flooded the zone with these empty decoys. The antibodies are overwhelmingly likely to bind to the useless spheres and filaments. They waste all their ammunition on the decoys, allowing the actual infectious Dane particles to slip past the blockade and infect the liver.
That is brilliant. It's like a fighter jet releasing flares to distract heat-seeking missiles.
Exactly. It is a literal smokescreen for the immune system.
Yeah.
And clinically, this is important because these decoys are what we detect when we measure the surface antigen in the lab. There is so much of it, it's very easy to find.
That makes a lot of sense. Now, let's talk about the engine under the hood, the replication. You said it's a DNA virus, but Murray mentions reverse transcriptase. I thought that was strictly an HIV thing.
It is the hallmark of retroviruses like HIV. But hepatitis B has a bit of an identity crisis. It uses it too.
How does that work?
Okay, stick with me here. The HBV genome is weird. It's a circular DNA, but it's only partially double-stranded. It literally has a gap in it. When the virus enters the nucleus, the host enzymes look at this sloppy DNA and say, whoa, we need to fix this. So they repair the gap to form a cccDNA covalently closed circular DNA.
cccDNA.
Got it. Think of the cccDNA as the master press. It sits inside the safe house of the nucleus and it does not leave. It acts as the template. The host cell transcribes this DNA into RNA.
Okay, standard central dogma so far, DNA to RNA.
But here is the twist. This RNA moves out to the cytoplasm, gets packaged into a capsid, and then the virus uses its own enzyme reverse transcriptase to turn that RNA back into DNA inside the capsid.
So it goes DNA to RNA back to DNA.
Exactly. It destroys the RNA template as it builds the new DNA strand. So the final virus particle contains DNA that was born from RNA, which was born from a DNA master copy.
That is incredibly convoluted, but why does it actually matter to you as a doctor?
Two main reasons. One, because it uses reverse transcriptase, we can use some HIV drugs like tenofovir and entecavir to treat hepatitis B.
Oh, that's handy.
It is. But two, and this is the big one, that cccDNA master press in the nucleus, it is incredibly stable. It's like a read-only memory archive. Even if we kill all the active virus in the blood, that archive is still sitting in the library of your liver cells, just waiting. This is why we can't easily cure hepatitis B. We can usually only suppress it.
That explains so much about chronic management. Okay, let's tackle the part that makes every medical student sweat. Hepatitis B serology, the alphabet soup. We've got S, C, and E antigens and antibodies.
It is a total rite of passage.
Yeah.
But we can simplify it. We have three main systems. Let's role play this. You would be the clinician looking at the lab report.
Okay, I am ready. Hit me.
First, the surface. HBsAg, hepatitis B surface antigen.
This is the coat, the flares, the decoys.
Right. Think of HBsAg as the presence marker. If it is positive, the virus is in the building. The patient is infected. It doesn't tell you if it's new or old, just that it's currently there. If it stays positive for more than 6 months, we define that as chronic infection.
Okay. Presence. And its partner, anti-HBs.
Anti-HBs is the surface antibody.
Mm-hmm.
This is the victory marker. It neutralizes the virus. If a patient is positive for anti-HBs, it means they are immune. They've either beaten the infection and recovered, or they have been vaccinated.
So HBsAg is I have it, anti-HBs is I won. What about the core, HBsAg?
The core antigen HBsAg is deep inside the virus particle. You generally do not detect the antigen itself in the blood because it's wrapped up in the coat, but you do detect the antibody anti-HBc. This is the history marker.
The history marker.
Yes.
Yeah.
Anti-HBc tells you if the immune system has ever seen the real virus. And it comes in two flavors, IgM and IgG.
Back to the timeline.
Right. IgM anti-HBc is the first antibody to appear. It indicates recent acute infection.
Wait, hold on. I need to push back on this because this is where everyone gets confused. You said surface antibody means victory. But in the textbooks, there's this gap called the window period. The surface antigen disappears, but the surface antibody hasn't shown up yet on the lab test. So if I test the patient then, both are negative.
Ah, the dreaded window period. Yes, that is correct.
But if the surface antigen is gone, shouldn't the antibody be there? Where is it hiding?
It is there, but it's invisible to the test. It's bound up. Imagine a battlefield where every single enemy soldier, the antigen, is grappling with one of your soldiers, the antibody. They cancel each other out. They form immune complexes and precipitate out of the solution.
So the lab test can't see them because they're literally locked in combat.
Correct. The lab reports negative for both. So in this window, what is left? The only evidence that the war is happening is the IgM anti-HBc.
So the IgM core antibody is the only witness left at the crime scene for that brief moment.
Exactly. It is a vital clue during the window period. Eventually, the IgG anti-HDC appears, which means the infection is old history.
So here is the million-dollar question for the exams. How do you tell the difference between someone who was vaccinated and someone who had the virus and recovered? Because both are going to have the victory marker anti-HBs.
This is the crucial differentiator. Think about the vaccine. What is it? It's a recombinant subunit vaccine. It contains only the surface antigen. It's literally just the coat. It does not contain the core.
Oh, so a vaccinated person has never seen the core.
Exactly. So a vaccinated person will have anti-HBs, the victory marker, but they will be negative for anti-HBc, the history marker.
Got it. But a person who had the real virus saw the whole thing, surface and core, so they will have anti-HBs and anti-HBc.
Precisely. Vaccinated equals surface antibody only. Natural immunity equals surface antibody plus core antibody.
Perfect. That leaves one more letter. EHBA.
The E stands for envelope technically, but for your memory, think E for explosive replication or excreted. HBA is a marker of high infectivity. It correlates with high viral loads. If a patient is HBA positive, they are shedding massive amounts of virus. They are highly contagious.
So if I accidentally stick myself with a needle from an HBA positive patient, I'm in much bigger trouble than if they're HBA negative.
Significantly higher risk, yes. It indicates the virus is replicating wildly.
Okay, let's talk about outcomes. You mentioned earlier that age plays a huge role in hepatitis B.
It does, and it's very counterintuitive. We usually think babies are more vulnerable to severe disease. With Hep B, it's the opposite regarding symptoms, but worse regarding long-term outcome.
Unpack that.
Remember, the liver damage is caused by the SWAT team, the immune system fighting the virus. Adults have a strong, mature immune system, so when an adult gets Hep B, they fight hard. They get severe acute symptoms, jaundice, fever. But because they fight hard, 95% of adults will win. They clear the virus and develop immunity.
High drama, good ending.
Exactly.
Yeah.
But neonates, their immune system is immature. It's built to tolerate things, to not overreact to every new protein it sees. So if a baby catches Hep B, usually from mom during birth, the baby's immune system looks at the virus and essentially says, you're probably fine. It tolerates the virus.
So no SWAT team.
No SWAT team. The baby rarely gets jaundice. They don't look sick at all. But because they don't fight, they don't clear it. The virus sets up that cccDNA master press and never leaves. 90% of infected infants become chronic carriers.
That is heartbreaking. So the baby seems fine, but the virus is setting up a permanent residence that will cause massive damage for decades.
That leads to the long-term sequelae. Chronic Hep B is a major cause of cirrhosis and, uniquely, primary hepatocellular carcinoma, or PHC.
Now, Hep C also causes cancer, but Hep B is different, right? It's a true oncogenic virus.
Yes. Hep B is a DNA virus. It can physically integrate its genome into the human host DNA. We call this insertional mutagenesis. It can hop into your genome and disrupt a tumor suppressor gene or land right next to an oncogene and turn it on.
So it's physically rewriting the cell's genetic code to promote cancer?
Correct. Hep C, being an RNA virus, can't integrate. Hep C causes cancer largely through chronic inflammation and regeneration, the constant cycle of damage and repair, which eventually causes a mutation. Hep B does that, plus the direct DNA integration. That's why the Hep B vaccine is essentially the world's first anti-cancer vaccine.
That is a powerful way to frame it. Okay, that's B, the complex chameleon. Decoys, reverse transcriptase, master press, age-dependent outcomes. Now let's move to the other major chronic player, Hepatitis C, the chronic shapeshifter.
Hepatitis C is a flavivirus. It's an enveloped, positive-sense RNA virus. And if Hep B is the master of decoys, Hep C is the absolute master of mutations.
This is the one that accounts for most of the non-A, non-B hepatitis from the old days, right?
Right. Before 1990, if you got a blood transfusion, you had a significant risk of getting this mystery hepatitis.
Yeah.
We now know that was HCV.
Why do you call it a shapeshifter?
It comes down to its enzyme, the RNA-dependent RNA polymerase. This is the machine that copies the virus. In most organisms, and even many viruses, there is a proofreading mechanism, a backspace key. If the enzyme makes a typo, it goes back and fixes it.
Makes sense. You want an accurate copy of your blueprints.
Usually. But HCV lacks a backspace key. It is incredibly sloppy. It copies the genome at a furious rate, literally trillions of particles a day, and introduces random mutations constantly.
But isn't that risky? If I typed a paper and made a typo in every sentence, it would be unreadable.
Most of the mutations are unreadable. They create broken viruses that just die.
Mm.
But purely by chance, a few of those typos change the shape of the envelope proteins, E1 and E2, just enough.
Just enough for what?
Just enough that the neutralizing antibody can't lock onto it anymore. Imagine the immune system makes a key to fit the lock. By the time the key is ready and deployed, the virus has changed the lock.
It's a moving target.
Exactly. This leads to the concept of quasi-species. In a single patient, you don't just have one homogeneous strain of HCV. You have a swarm of closely related variants. The immune system is constantly playing catch-up and just losing.
And that explains why we don't have a vaccine for C.
Exactly. We can't make a vaccine against a target that changes its face every single day. And it also explains the clinical course. C is for chronic.
What are the actual numbers on that?
Unlike Hep B adults who clear it 95% of the time, with Hep C, the immune system just gets exhausted by this constant shape-shifting. 70 to 90% of patients become chronic carriers.
That is a huge difference. High chronicity. And clinically, it's often quieter than B, right?
Very insidious. Acute infection is usually asymptomatic or very mild. People can walk around for 20 or 30 years not knowing they have it, while the virus slowly scars their liver. It's often called the silent epidemic, especially among baby boomers born between 1945 and 1965. And
eventually that scarring leads to cirrhosis and cancer.
Yes. Cirrhosis in about 20% of chronic carriers and carcinoma in about 5%. And alcohol consumption massively accelerates this process.
Now, here is where the story gets surprisingly hopeful. You said Hep B is managed, not cured, because of that nuclear cccDNA. But Hep C?
Hep C has undergone a total therapeutic revolution in the last decade. We used to treat it with interferon and ribavirin. It was toxic. Patients felt like they had the flu for a year, and it didn't even work very often.
The dark ages of hepatology.
Truly. But now we have DAAs, direct-acting antivirals. These are designer drugs that target specific viral proteins. We have protease inhibitors, phosphoprotein inhibitors, polymerase inhibitors.
How do they work?
I think of the virus as a machine with gears. These drugs are like jamming a wrench directly into the gears. They stop the protease from cutting the proteins, or they stop the polymerase from copying the RNA.
And because Hep C is an RNA virus,
It stays in the cytoplasm. It does not have a DNA master copy hiding in the nucleus like Hep B. It has no archive. So if you jam the gears and stop replication for 8 to 12 weeks, the existing virus just dies out, and there is no backup copy to restart the infection.
so the whole house of cards collapses.
Completely. We can now achieve what we call SVR, sustained virologic response, in over 95% of hepatitis C patients with just a course of pills.
That is one of the greatest triumphs of modern medicine, going from a mystery virus to a literal cure in roughly 30 years.
It really is remarkable.
Let's briefly touch on hepatitis G.
HGV or GBVC. It's another flavivirus very similar to HCV. It's blood-borne. We find it in blood donors. It establishes chronic viremia. But honestly, we aren't really sure it causes significant liver disease,
So it's a virus looking for a disease.
basically. In fact, there's some fascinating evidence that in HIV patients, co-infection with HGV might actually be protective and slow down HIV progression by interfering with viral entry. But for the purpose of the liver, it's not a major clinical player like D or C.
Okay, so G is the weird cousin. Let's move to D, hepatitis D, the defective parasite.
This is a fascinating biological entity. Hepatitis D is the smallest human pathogen. It's basically a viroid-like circular RNA, but it has a fatal flaw. It cannot make its own envelope.
It's incomplete?
It's naked in a bad way.
Yeah.
To be infectious, it needs a coat to enter a hepatocyte. And specifically, it needs the HBs AG coat.
Wait, it steals Hep B's coat.
It steals Hep B's coat.
Ass.
This means you strictly cannot have hepatitis D unless you also have hepatitis B. It is an obligate parasite of a parasite. Even fleas have fleas.
This creates two distinct clinical scenarios that are super high yield for exams. Scenario A is co-infection.
Co-infection is when a patient gets Hep B and Hep D at the exact same time. For example, an IV drug user shares a needle that has both viruses on it.
So both enter the body together.
Right. They both cause an acute hepatitis. It can be severe because you have two viruses attacking the liver simultaneously. But here's the key. Since the patient is an adult, usually what happens to the Hep B?
The adult immune system clears Hep B 95% of the time.
Correct. And if Hep B dies...
Hep D dies, it loses its supplier.
Exactly. So generally, co-infection resolves. It's a stormy course, but the patient usually clears both viruses.
Okay, contrast that with scenario B, superinfection.
This is the nightmare scenario.
Yeah.
You have a patient who is already a chronic carrier of hepatitis B.
Yeah.
They have the HBs AG factory running 24/7. Their liver is just full of surface antigen.
The factory is open for business.
Then they catch Hep D later. The Hep D enters and finds an absolute paradise. Unlimited coats. It doesn't have to wait for Hep B to set up. The infrastructure is already there. Hep D replicates explosively.
And what does that look like clinically?
It looks like a rapid, severe deterioration. A stable Hep B patient suddenly gets fulminant hepatitis. The risk of cirrhosis accelerates dramatically. It's highly dangerous.
So co-infection equals bad, but usually clears. Superinfection equals very bad, chronic, dangerous.
Correct. And prevention. If you vaccinate against B, you automatically protect against D. No B, no D.
Two birds, one stone. Finally, let's wrap up the alphabet with hepatitis E, the enteric enigma.
Hepatitis E is remarkably similar to hepatitis A. It's a naked RNA virus. It's transmitted fecal orally, usually through contaminated water. It causes large epidemics in developing countries.
So E for enteric, vowels and bowels.
Yes, and typically it's acute and self-limiting, just like A. But there is one massive tragic exception that you absolutely must know for exams.
The pregnant exception.
Correct. For reasons we still don't fully understand, likely related to the shift in immune status during pregnancy, hepatitis E is incredibly dangerous for pregnant women. If a pregnant woman gets Hep E, particularly in the third trimester, the mortality rate can be up to 20%.
20%. That is shockingly high for a self-limiting virus.
It causes fulminant hepatitis, which is rapid liver failure. So the mnemonic here is E for expectant mothers. Any vignette involving a pregnant woman with liver failure, especially returning from an endemic area like India or Mexico, you must think hepatitis E.
And in the developed world, is it here?
It is, but it's slightly different. It's a zoonosis here. We see it linked to undercooked pork or game meat.
So A is shellfish, E is pork and water.
A good rule of thumb.
Wow. We have covered the whole alphabet. It really is a strategic game between the host and the pathogen. Let's bring it all back together.
It is a massive chess match. You have A and E, the hit-and-run squad that come in through the gut, hit the liver hard and fast, and leave. A is dangerous for the community due to asymptomatic kids. E is dangerous for pregnant women. Then you have B, the master strategist, a DNA virus that uses decoys to confuse the enemy and hides its genetic code in the nuclear cccDNA. It plays the long game, especially in babies who can't fight back.
Right.
You have C, the shapeshifter, an RNA virus that survives by constantly changing its appearance via a sloppy polymerase, leading to high chronicity, but thanks to cytoplasmic replication, is now totally vulnerable to our cures. And D, the opportunist, a defective virus that piggybacks on B to cause maximum destruction.
It's a diverse crew, all after the exact same prize. Before we hit the rapid-fire questions, I want to leave you with a provocative thought based on what we just discussed. We have a cure for C, but not B.
It really all comes down to location. Hep C is in the cytoplasm, Hep B is in the nucleus. That cccDNA minichromosome is the final frontier. Until we figure out how to scrub that DNA out of the host nucleus without actually killing the cell, Hep B will remain a managed disease rather than a cured one.
The dormant sleeper agent. Fascinating stuff. Okay, are you ready to test our learners?
I am ready. Let's do it.
Rapid fire, single best answer. I'll give you a snapshot, give you a couple seconds to think, and we will reason through the diagnosis together. Question 1. A 27-year-old intravenous drug user presents with nausea and jaundice. Serology shows HB-Ag positive and anti-HBC IgM positive. Okay, let me think about that one. So we have HBs-Ag positive, which means the virus is there. But is it new or old?
Look at the antibody. It's IgM. IgM is the Marine, the first responder. Yeah. Since the first responder is still there, this must be a new attack. So the diagnosis is acute hepatitis B.
Question 2. A 30-year-old pregnant woman returns from a trip to India and presents with rapid onset permanent liver failure. Okay, the travel points to something infectious, but the key here is the pregnancy and the severity.
Exactly. Hep A doesn't typically kill pregnant women like this. The high mortality in the third trimester is the signature of the enteric enigma. Diagnosis, hepatitis E.
Question 3. A patient complains of chronic fatigue. They test positive for anti-HCV. They ask you why there isn't a vaccine for this yet. What is the biological reason?
Good question.
We talked about this. It's the shape-shifting, right?
Right, but specifically it's the mechanism. The virus lacks proofreading. The answer is the error-prone RNA polymerase generating quasi-species. The virus changes its code faster than we can design a vaccine for it.
Question 4. You are reviewing a lab report. The patient has anti-HBs positive and IgG anti-HBC positive. HBs-Ag is negative.
Okay.
Let's see. Anti-HBs means victory. They are immune. But the IgG anti-HBC means they have a history.
Right. They have antibodies to the core. The vaccine doesn't have the core, so they must have seen the whole virus. Diagnosis, resolve past infection, which means natural immunity.
Question 5. Compare that to this one. Patient has anti-HB is positive, but anti-HBC is negative. Victory marker is present, but history marker is absent.
They have the surface antibody, but never saw the core. That means they only saw the protein coat, not the whole virus. Diagnosis, vaccinated.
Question 6. A patient with known stable chronic hepatitis B suddenly gets rapidly worse, with spiking liver enzymes and severe jaundice. What happened?
Oh, this is a bad day.
The factory was open, chronic B, and suddenly it went into overdrive.
This suggests a new agent entered the mix and utilized the HBs-Ag. Answer, HDV superinfection.
Fantastic. If you want to solidify this, go open Murray chapter 55, look at table 55.1 for the virus comparison, and table 55.2 for the serology. That serology table is absolute gold for exam.
Absolutely. Visualizing the timeline of antigens and antibodies is the best way to make it stick.
Thanks for jumping into this deep dive with us today. Wash your hands, cook your shellfish thoroughly, and we'll catch you on the next one.
Stay curious.